o'clock here Eastern time. We're going to go ahead and get started to make sure our presenters all have the ample opportunity to give their talks today. So welcome to the general session of the Waterbird Society. We've got 5 presenters today. 4 of them are students. So big welcome to all 5 of them. Danielle, at some point today, will put the nomination form for students in the chat box. So please remember to take notes and nominate students, speakers, presenters as you see them. So first up today, we have our first student who is Jill Tengaris. So Jill, if you want to go ahead and share your screen, you can go ahead and get started.
All right. Thanks for the introduction. If you can't hear me, someone should let me know.
You sound good.
Okay, thank you.
And your slides look good.
All right, cool. All right, well, thanks. Hi, everyone. So my name is Jill Tengaris, and I'm a master's student working with Dawn Lyons at Oregon State University, where I'm studying the breeding ecology of Aleutian terns in the Kodiak Archipelago up in Alaska. So today I'm going to talk about some preliminary results stemming from my research studying Aleutian tern chick diet. So let's get started. So first, I just thought I'd give a brief overview of some life history traits of this species, since some of you watching today might not be very familiar with them. So this species of tern only breeds in coastal areas of Alaska and the Russian Far East, and Aleutian terns migrate to their wintering grounds in Indonesia via the East Asian Australasian flyway. And just a side note, this is the only species in its genus that isn't strictly tropical. And also, Aleutian terns have a breeding ecology more like black terns than other species in their genus, so they're kind of a little bit of an oddball with their closest relatives. So they can nest in colonies or semi-colonially, and they usually exhibit a dispersed nesting strategy. And also these colonies can be monospecific or mixed species. In Alaska, we often see them nesting with Arctic terns and meagulls, and they usually only lay 1 to 2 eggs per clutch. So you may be wondering why I'm interested in Aleutian terns. Well, in 2015, there was a paper published by Renner et al. That estimated that the Alaskan breeding population of Aleutian terns had declined by 92.9% in the previous 33 years, which is obviously very alarming and concerning. And this paper highlighted how little information exists about Aleutian tern biology, and that there needs to be some consistent and standardized monitoring of the species across the state to really understand what's going on. So this kind of kickstarted a lot of Aleutian tern research across Alaska, including the research that I'm going to talk about today. So one of the data gaps in Aleutian tern work is diet. We don't really have much information about what the adults or the chicks eat. So my thesis research is looking at both chick and adult diet, but today I'm mainly going to focus on chick diet. So for species like Aleutian terns, understanding chick diet could help us interpret how changing ocean conditions are affecting availability for the species and potentially contributing to this decline. So because Aleutian terns tend to nest in low densities and often in vegetated areas, using the traditional method of documenting feeding events by an observer sitting in a blind wasn't really going to work for the species at our colonies. So instead, we decided to use Reconnex trail cameras placed at about 1 meter from the nest to document feeding events. Aleutian terns adults carry fish in their bills, so hopefully the camera pictures would allow us to identify species. So our cameras were programmed to take a photo once every 2 minutes, and also to take a burst of 3 images whenever the camera was motion triggered. So in this picture on the right side of the slide, here you can see one of our cameras, and it's pointed at a nest bowl where there's a little chick and an egg. And as you can see, they're pretty beautifully camouflaged, and these nests are not easy to find on the landscape. So all of the research this research was conducted in the Kodiak Archipelago in Alaska, which is located in the Gulf of Alaska. And in my opinion, this is a great place to study Aleutian terns. There's 24 documented colony sites across the archipelago, and some of these sites even have a little bit of historical data, which is kind of unique for the species. So I would like to note that not all of these colony sites are active within a given year. Some of them haven't been active for decades, and some of them, they're active every few years, but we have options here, and that's pretty nice for this species. Also, we have 5 colony sites on the road system that have been active within the past decade or so, and these colonies are really easy to access. We can drive right to them, which is pretty unique and allows us to do some intensive research that isn't really possible at other sites across Alaska. So today I'm going to share some of my data from 2017 through 2019. I was able to conduct fieldwork safely in 2020, luckily, but that data isn't quite ready, so I'm not going to talk about it very much. So I've subsetted all the camera data down to just nests that successfully hatched chicks for this analysis. There's a ton of other data out there, but that's going to be used for other projects. So I ended up with chick feeding data from 4 different colonies, 3 colonies that were on the Kodiak Road system, including Middle Bay, Callison Bay, and Burton Ranch, and 1 that was down at the southern end of Kodiak Island called Anchor Cove. So here is just a quick summary of how many feedings we were able to document using Nest cameras. I did add the 2020 data here just as a teaser. So we concluded 4 years of fieldwork with a total of 1,334 feedings, which I was very excited about. I did not expect this many when we started. So we really did learn quite a bit here. So let's dive into this data a little bit farther. So first I want to talk about feeding rates. From all of the feeding events from 2017 through 2019, I filtered out feedings that happened within the first 3 days after the first chick hatched, and only feedings that happened when there was 1 chick at a nest. So we only used the first 3 days for this analysis because as Aleutian tern chicks get older, they tend to wander away from the nest. And our chance of the chick being fed outside of the field of view of the camera increases a lot. Aleutian tern chicks don't like to sit still for very long, so they definitely like to wander around. And we only used nests with 1 chick just to eliminate some variation in feeding rate that could be influenced by having a second hungry chick present at the nest. So also just a quick note that for this presentation, I've included all fish that were brought to the nest and presented to the chick. I can't confirm that every single one of these fish was consumed by the chick and not the adult, or it was dropped on the ground or something, but that is something I'm hoping to look at a little bit more in my actual thesis. So as you can see in the table here on this slide, the median number of chick feedings in 2017 was 2, which appeared to be lower than what we were seeing in 2018 and 2019. So due to the non-normal distribution of the feedings per day data, I used a Kruskal-Wallis test, which was then followed up by a Dunn test to investigate any difference in feedings per day between these 3 years. So the Dunn test produced a significant p-value at an alpha of 0.05. So we concluded that there's a higher feeding rate in 2019 than 2017. The comparison in 2017 and 2018 doesn't appear to be significant, even though the median feedings were similar and the range of the number of feedings per day was similar, but it might be because there was a pretty small sample size. So next, let's look at diet composition. So I have a few pictures from the Nest camera so you can get a feel of what some of this data looks like. So this is a really great picture, and I wish that all of them looked like this. It would make my life a lot easier. But here we get a really great view of this fish, and with that big yellow eye, and we know this is a kelp greenling species. So perfect. Similarly here, this is definitely a sand lance. People describe them as a noodle. They're just long and thin, so they're easy to identify, thankfully. But this picture is much more normal, I would say. We know it's a fish, but we don't get a good view of any traits of it to really get an ID on this. So unfortunately, this just has to remain an unknown fish. So I'm sure you can imagine that IDing these fish can be very challenging. So 32% of our feeding events from 2017 through 2019 were unidentifiable, but I'm still pretty happy with how many we were able to identify, especially because in this pie chart, the unidentifiable prey includes unknown fish, where we at least know it was a fish and not an invertebrate per se. So we still have some data that we can use there. So for the rest of the analysis that I'm going to show, I removed all of the unknowns just to help make the picture a little bit more clear. So let's take a look at this data. So I know this is kind of busy. So down here on the x-axis, we have the 4 colonies where we collected chick feeding data, and plus 1 column that just summarizes all of the data. And on the y-axis, we have the proportion of the diet. So let's look at these 3 bars in the middle here. These represent the 3 road system colonies. And you'll notice that there's a lot of dark green up here. And these are the hexagrammid species, or the greenling species. So this is the fish with the yellow eye that we saw on the first example picture. And below the green, there's this red. And this is Pacific sand lance, which is a super common prey species for marine predators in this system. So now let's look over here at the first bar at Anchor Cove, this colony that's not on the road system, and it's at the southern end of the island. Here we see a lot of sand lance again, a lot of red, a little bit of green, but a lot of orange. And the orange are salmon. And it looks like mainly chum and pink salmon, as far as I can tell, but I'm not a fish expert. Let's keep going for diet composition. So this is just year now on the x-axis with proportion of diet. On the y, just ignore this white space for a second. And we can see between years, all 3 years have a lot of hexagrammids, a lot of sand lance, a little bit of salmon. And 2017 was a little bit interesting that there was quite a few gaddids, mainly Pacific cod up here. So now I'd like to add 2 bars of data that I extracted from a report published in 1983 as part of the Outer Continental Shelf Environmental Assessment Program. Though the sample sizes are really small for these 2 years, I think that they can illustrate at least that osmerids, mainly capelin in this instance, used to be much more numerous in the diet of Aleutian tern chicks in Kodiak. And something happened between then and now that they are no longer present. Aleutian tern colonies were bigger back then, which kind of makes me suspicious about maybe this is influencing Aleutian tern productivity and helping contribute to the decline that we're seeing. And that is something that we're definitely trying to look into. All right. Oop, and there's a capelin. All right, so let's go on to conclusions. So first, we found that chick feeding rates varied across years. And we kind of assumed or suspect that prey availability, plays a role in this. But unfortunately, this is not a question that I'm attempting to address. I didn't talk about it at all in this presentation, but I'm planning at looking at how feeding rates and diet composition relate back to nesting success. So for example, let's think back to that first example of the feeding rates where we saw that 2017 was significantly lower than 2019. So just to give you some context to that, that was kind of what we expected to see. So 2017 was a horrendous nesting year in Kodiak. Very few terns attempted to nest at all. I only saw 4 fledglings over the entire archipelago. That's pretty bad, but also kind of normal for the species in a way. But in 2019, when the feeding rate was higher, a lot of terns started to nest. At one colony on the road system, we had over 35 fledglings, which is phenomenal. I know 35 doesn't really sound phenomenal, but I promise you it was great. So I'm hoping to investigate more how these metrics of nesting success can relate back to what we're seeing from this diet data. All right, so then conclusion number 2, we saw that Pacific sand lance and hexagrammid species were the most common diet items in 2017 through 2019. And I suspect that once I add the 2020 data, that this trend will hold. It seems like it's pretty consistent so far with the previous 3 years. And for number 3 down here, we found that osmerids aren't important in chick diet anymore. And we don't really know the implications of this, but there definitely was a shift in the diet, it seems. And we're hoping to investigate that a little bit more. So before I conclude, I would just like to give a little preview of some other work and some next steps with concerning evolution-turned diet. So another part of my thesis research is looking into the pre-Lang diet of female terns using stable isotope analysis of eggshell membranes. So I just got my first batch of preliminary data back, and I just thought I would share it briefly as a teaser. So here we have the carbon isotope values on the x-axis and nitrogen isotope values on the y-axis. And I have the data group by year. And you can see that these isotopic niche ellipses overlap for 18 to 20. But this 2017 ellipse is out here doing its own thing. It seems pretty different from the following 3 years. And that kind of is consistent with what we saw for feeding rate. So this isn't all the data. I still have some eggshells that need to be processed, but I'm hoping to present on this at the Pacific Seabird Group meeting in this winter. I think it's February. So if you're interested, stay tuned until then. So with that, there's a lot of people to thank. As those of you know who work with camera data, it's a lot to process, and I've been lucky to have a lot of really great help. And big thanks to Kirsten, Adam, and Alyssa for helping with Fish ID. I really appreciate all of their help. So with that, I think I have a couple minutes for questions.
You sure do. Thanks very much, Jill. Audience, feel free to unmute yourself, turn your video on if you'd like to ask a question, or write in the chat. And I see Erica Knoll has already left a question, which is, Jill, did you get a fish biologist to look at the photos of your prey? They might be able to help identify some of those unknowns.
Yeah, I've worked with a few people, the ones that I have issues with, and this isn't final. There's definitely some in the unknown categories that I'm hoping to get some other people to look at. So if there's any fish experts in the crowd who want to help, I would be happy to have your help. So just feel free to contact me.
Emily Johnson asks, have you looked at if feeding rate and nutritional value of the fish are related?
I haven't yet, but Emily, that is a great question. I think there is some information about the nutritional value of these fish, obviously, in the literature, and that is something I would like to look at to also pair with the isotope values that I'm still working on now. So hopefully, I still have a lot of work to do on my thesis, so some of these things can be incorporated in.
Jeff Spendelow asks, how frequently did you have to change batteries on your game cams?
It really depended. Some of the cameras were fine for like 3 weeks at a time. And then some, they would die quicker, and it really depended on how active the terns were and how close we had to put the camera to the nest based on the vegetation. As you can see, even from this slide that I have up right now, sometimes the vegetation's thick. And to get a camera in there that you can like see the nest and not just have vegetation in front of it, it has to be pretty close, and then the motion trigger goes off a lot. So it eats up the battery and the memory much quicker. So it depended. The road system cameras I checked weekly just to try to make sure that I didn't lose data because of batteries dying.
Just going to ask one more question because we're almost out of time, but you've got a lot of great comments here in the chat. The last question is from Matuis. Says, amazing talk. Were you able to recognize the sex of adults? No, unfortunately not. I wish we could. That would be very interesting. Thanks again, Jill. So We will have a discussion period at the end of this session, so if you want to stick around and ask general questions later, please stay through the 3 o'clock period. But next up, we have our second student, who is Bradley Wilkinson. So Bradley, if you'd like to go ahead and share your screen.
Perfect. Is that working for everyone?
Yep, thank you.
Excellent. Well, today I'll be talking about perfluoral alkyl substances in the eggs of brown pelicans from South Carolina. This is part of my dissertation research at Clemson University with Dr. Patrick Jodas. So as we know, seabirds are often considered to be effective sentinels of marine pollution, given their trophic position, as well as their ability to cover wide geographic areas. Brown pelicans in particular have a long history with anthropogenic contaminants, especially DDT and DDE. These two compounds caused eggshell thinnings in this species, which led to widespread reproductive failure. Brown pelicans were listed under the Endangered Species Act in 1970. Following bans of DDT and DDE, the species recovered and was delisted in 2009. I take all of this as evidence to be that brown pelicans are likely a good indicator for both legacy and perhaps novel compounds in nearshore environments, which is going to be setting up the rest of my talk. So perfluoral alkyl substances, or PFASs, have been manufactured since about the 1940s. Primarily, they're used in non-stick cookware, such as Teflon, for water and oil repellent textiles like Scotchgard, paper products, and also in firefighting foams. Despite having been produced since the 1940s, not a lot was known about them for a long time until just recently. As a result of some of this pioneering research, production bans are in place for some compounds starting in the early 2000s. However, there's a bunch of PFASs, and over 4,000 compounds classified as PFASs are still on the market. And in fact, the compounds that have been banned in the United States are still available abroad and can make their way into our ecosystems. So perfluorooctane sulfonate, or PFOS, and perfluorooctanoic acid, or PFOA, are the most common compounds found in wildlife and will be the ones that I'll be discussing. So PFASs are unfortunately very persistent in the environment. They have earned the moniker forever chemicals because of that fact. I included a quote here from Hood et al. 2006. It is widely recognized that PFSAs and PFCAs are persistent in the environment. To date, there is no evidence for biodegradation of these chemicals, which is quite frightening. In addition to their persistence, they are able to bioaccumulate and biomagnify. Unlike a lot of other chemical compounds, such as PCBs or PBDEs, which are lipophilic, PFASs bind to blood proteins, which means they concentrate in the liver, kidney, bile, and albumin. In addition to being a threat for wildlife, they're also a human safety concern, so much so that a blockbuster movie was actually recently released called Dark Waters, starring Mark Ruffalo, about PFAS in industry and its impact on human health. I haven't actually seen the movie, but I've heard it's good and somewhat educational as well. But in fact, estimates suggest that PFASs are detectable in the blood of over 99% of Americans. So in addition to being a wildlife concern, they're also a human health concern as well. My dissertation research takes place in Charleston, and over the past decade, there's been some really interesting and groundbreaking work about PFASs in the Charleston area. Certain studies have looked at fish and found very high levels in edible fish beyond what are safe thresholds for mammalian consumption. Estuarine sediments in Charleston have been found to contain really high levels of PFAS. And most interestingly, or perhaps tragically, bottlenose dolphins in Charleston have very high levels of PFAS. And one study, in fact, found blood levels of PFAS to be as high as those in occupationally exposed humans. So bottlenose dolphins have some of the highest levels of PFAS globally in any sort of marine mammal. A study that inspired this study that I'm currently doing was by Adams et al., looking at different subpopulations of bottlenose dolphins in the Charleston area. So the map on the bottom left shows Charleston Harbor with the Ashley, Cooper, and Wando rivers leading into it. The red are urban areas, and then the green and yellow are forest and agricultural land, and then the blue are estuarine areas. So they compared dolphins that were known to be sedentary within the harbor with those that were known to be sedentary in the Stono River estuary, which is approximately 10 kilometers to the southwest. And so they took blood samples from dolphins from those two areas and found that PFAS levels in dolphins that were resident within the harbor or one of those rivers had much more high concentrations of PFAS than those that were resident within the Stono River estuary. This inspired our work because we have pelican colonies that are both within the harbor and at distance from Charleston Harbor. And so one of our objectives was to see if we could replicate this spatial structure that was found in dolphins in another upper trophic level marine species, the brown pelican. So objective one was to determine PFAS levels in, like I said, another top marine predator from the Charleston region, the brown pelican, and to investigate the influence of urban proximity to PFAS concentrations in the eggs of those pelicans. So here's a map of our study system here. We have 3 colonies that are part of this study. The first is Castle Pinckney, which is located right in Charleston Harbor, just a stone's throw from the Ashley Cooper and Wando rivers. We have Bird Key Stono, which is at the mouth of the Stono River, which is in the heart of the Stono River estuary, much as in the dolphin paper. And we also have DeVoe Bank, which is another approximately 10 kilometers to the southwest of Bird Key Stono. So we have a close colony, a medium distance colony, and a relatively far away colony. We collected 36 eggs in total, 12 from each colony, in May 2019 at the start of incubation. We collected 1 egg per nest, and we floated to determine the freshness of the eggs. We only wanted eggs that were relatively recently laid. We didn't want any developing embryos. And then we made an effort to collect the first egg of every clutch based on the appearance of the eggs. So pelicans lay 3 eggs, and it's been shown that first laid eggs tend to have higher levels of contaminants than later laid eggs. So we made an effort to collect the first out of every clutch. I'm going to go relatively quickly through the methods, but briefly, whole eggs were refrigerated until analysis, and then we homogenized the contents of each egg using a bag mixer. We prepared samples following established protocols. If you have questions, I'd be more than happy to talk about those later. We extracted PFAS from each sample using wax cartridges and an ENVI carbon sorbent. And then those extracts were analyzed for PFAS homologs using high-performance liquid chromatography mass spectrometry. We also have GPS tracking data from adult pelicans from 2017 to 2020. We've so far captured 75 individuals and gotten usable tracking data from those adults. We extracted just the breeding season movements based on nest activity. I built a recursive algorithm in R to determine the frequency of nest visitation for each individual. And once those individuals stopped revisiting that nest, we decided that was the end of that individual's breeding season. So we removed the breeding season movements only. And then some individuals who do not migrate actually stay at the colony year round. And for those individuals, we imposed a cutoff of 90 days once eggs had been laid. And that corresponds to roughly the maximum amount of time needed to successfully raise chicks in this species. So after we had all of our locational fixes, we projected that onto a 1.5 kilometer by 1.5 kilometer grid. And then we took the upper quartile of grid cells and defined that as high use. So the top 25% of grid cells that contain the most number of locational fixes for both Castle Pinckney, Bird Key Stono, and DeVoe Bank. And we use that as a proxy for the local ranges during the breeding season for individuals from each colony. So getting into some results, we found 16 PFAS compounds in at least 50% of samples, which is quite a lot. Unsurprisingly, PFOS was the most abundant compound across all of our samples. And in fact, the levels of PFOS that we found are among the highest reported among seabird eggs globally. They ranged in our study from 48 to 546 nanograms per gram wet weight. And just for some perspective, anything above about 100 nanograms per gram wet weight is pretty significant. So the fact that we were finding eggs that had almost 600 nanograms per gram was really a stand-up number. Interestingly, colony profiles were largely the same. Of the 16 high-frequency compounds, only 4 showed any statistical differences between those colonies. So the figure on the right is a figure showing overall PFAS contamination separated out by colony. So the gray is DeVoe Bank, yellow is Castle Pinckney, and blue is Bird Key Stono. You can see that DeVoe Bank had overall the highest levels of concentrations and Bird Key had the lowest mean concentrations, but these were not significant. The concentrations were not significantly different between colonies. So in the background here is a map of our breeding season movements. The red grid cells represent high use areas for Castle Pinckney. The blue represent DeVoe Bank, and then that kind of greenish tan color represents Bird Key Stono. What I want you to see here are that Castle Pinckney and DeVoe Bank are very well spatially segregated during the breeding season. Individuals from Castle Pinckney will forage within Charleston Harbor, unsurprisingly, and they'll also frequently use the Ashley and the Cooper River, as well as areas to the north. DeVoe Bank, in contrast, forages almost exclusively to the southwest of the colony, sometimes at pretty great distance, getting into coastal Georgia even. And then individuals from Bird Key Stono forage right around their colony as well, but also use a lot of areas that are used by Castle Bank or Castle Pinckney birds and DeVoe Bank birds as well. So they're kind of a mix between those two colonies. However, the 4 compounds that we did have differences in didn't really reflect this, in my opinion. So that figure, the inset figure, are the 4 compounds for which we had differences: FOSA, PFPEA, PFOA, and PFNA. And the takeaway that I want you to see for this are that of the compounds for which we found differences, 3 of them had the highest levels at DeVoe Bank, followed by Castle Pinckney, and then Bird Key was the quote-unquote least contaminated for those compounds, which is kind of a different pattern than what we were expecting. We expected Castle Pinckney birds to have the highest levels of concentration, followed by Bird Key, followed by DeVoe Bank being the cleanest, but that's clearly not what we saw. So I'm going to spend kind of the rest of the talk discussing where these contaminant sources may be coming from. So that's the big question: where are brown pelicans acquiring these contaminant loads? It's important to know that egg contaminants reflect the energy sources that were used in the development of those eggs. So kind of a key question in reproductive biology is determining if a species is a capital or an income breeder. So capital breeders use the energy that they have developed on the wintering grounds or during migration, like fat reserved energy to develop their eggs, whereas income breeders use locally obtained resources during the pre-breeding season to develop their eggs. So just remember that capital breeders, you're thinking migration or wintering sources, versus income breeders, you're thinking about locally obtained sources at the breeding colony. But it's also important to note that these are 2 endpoints on a spectrum. So on one hand, you have capital breeding, and on the other hand, you have income breeding. But in reality, most individuals fall somewhere in between purely capital or purely income. And it's unclear where along the spectrum pelicans fall, but I'm going to consider both capital and income sources. So if we're considering exogenous sources for egg development or income, we're going to be considering sources in and around the Charleston area. Charleston sources could include military bases where they use a lot of firefighting foams. A good example is the Joint Base Charleston Air Force Base, which has documented to release a lot of firefighting foams during training exercises into the Ashley River. Wastewater treatment plants could also contribute, as well as being shed from container ships. And there's also a lot of industrial activity centered along the Ashley and Cooper rivers. So this figure that I've included here is a map of the Charleston Harbor, and each one of those black kind of square checkmark looking boxes represent an end pipe from either a wastewater treatment plant or an industrial center that is flowing directly into the body of water. So every one of those marks on the Cooper River represents effluent that's flowing, like I said, from either industry or wastewater treatment directly into that river system. There's clearly a lot of industrial activity going along in those areas, which could be offloading PFAS into the environment. What's interesting is that we also found high levels of FOSA in our pelican eggs, which suggest recent and also continued exposure. FOSA is a precursor compound to PFAS. So when organisms intake FOSA, they are able to biotransform it into PFAS at a certain rate. And the fact that we saw really high levels of FOSA indicates that Our pelicans are intaking fossa at such an amount as to swamp the metabolic capabilities of the individuals to transform fossa into PFAS. So this suggests that they're intaking fossa currently, which is a big deal because fossa and PFAS were both part of the 2001 production bans. And so that indicates that even though both of those chemicals were banned for production in our area, they're still making their way into the environment. Of course, we also need to consider capital sources or endogenous sources of contaminants. And this is really tricky for pelicans because pelicans are facultative migrants. That means that in any given year, some individuals will stay around the colony year-round, whereas the other part of the population may migrate. Individuals can migrate only a couple hundred kilometers. They can migrate over 1,000 kilometers. It's extremely variable between individuals and also between colonies. So linking contamination with non-breeding movements is a real challenge. We've tracked pelicans from South Carolina all the way up to Chesapeake Bay, Virginia, down to Cuba, into Mexico and the Florida Bay, and they could be picking up PFAS contamination at any number of those sites, pretty much anywhere along the South and Mid-Atlantic Bight and the Caribbean as well. Some studies suggest that non-point PFAS contamination along the Atlantic coast may really be significant, so they might not necessarily be at some sort of industrial center to be picking up these contaminants. It could just be locally present in the environment, but that's, like I said, going to be very difficult to pinpoint exactly. The main outcome of concern for this high level of PFAS in our population is reduced hatchability of eggs. A study by Custer et al. Reported decreased hatching in tree swallows at about 150 nanograms per gram's wet weight of PFAS, and a 50% reduction in hatching at about 500 nanograms per gram wet weight of PFAS. In our study, 5 out of the 36 pelican eggs had concentrations of PFAS above that first threshold, and 2 out of the 36 had concentrations above that second threshold, which could indicate that our pelicans may be undergoing maybe experiencing reduced hatchability in our population. It's important to note that that study was done on tree swallows, and there's very different biokinematics between tree swallows and brown pelicans. So research would need to be done to determine hatching effects for our species in particular, as well as any sort of population level impacts would need to be further studied. We're providing here just more of a baseline analysis for what we're seeing currently, and we hope that future studies in different areas and also potentially at different times would add to the collective body of knowledge about PFAS contamination in this population. So I'd like to thank a couple people for field assistance, lab assistance, and also project advisement. And with that, I can take a couple questions if I have some time.
Thanks, Bradley. We do have about 2 or 3 minutes for questions if anyone wants to chime in or write in the chat box. I can chime in while we're waiting. So I'm definitely not a contaminants biologist. This is a pretty naive question, but I've seen a lot of literature on contaminants in gulls. And just wondering sort of the interchangeability. You touched on this at the end of your talk, but the interchangeability of species when you're looking at contaminants. Is it just a matter of studying whichever species is most locally convenient, or are some species really better indicators than others of contaminants?
Yeah, I think there certainly are a lot of species-specific like biokinetics that are at play. Gulls, especially like in the Great Lakes area, have been studied quite a bit. Gulls are tricky because they integrate both terrestrial and marine systems. So part of the reason we chose pelicans is because we were looking for just like a near shore exclusive signal. Gulls, you're having intake from a lot of, like I said, a lot of different inputs. The big thing for comparison's sake, I guess, between published studies is looking at the tissues that were examined. So some people looking at PFAS, dissect out livers, some are looking in blood, some are looking like we did in eggs. And it's really difficult to compare across tissues and across species. It's much more easy to compare either within a species looking at different tissues or the same tissue in different species, if that makes sense.
Yes, thank you. Don Lyons asks, could you get at the capital income question by comparing first eggs to third eggs?
Potentially, yeah. So if we were looking, for example, at stable isotopes of first versus third eggs, you might be able to see that first laid eggs are using more capital than later laid eggs. I believe pelicans lay about 1 egg per day. I don't know the magnitude of change that you would see in between like a first or third egg, but it's certainly possible. I think another way you could get at that would be to look at eggs that are laid across the breeding season. So we took eggs all at once in May, but if you were to collect eggs from early season breeders versus mid-season breeders versus late season breeders, it's likely you'd see a decreasing amount of capital in those clutches throughout the breeding season, and you might be able to get at contaminant sources that way.
Thank you, Bradley. It is time to move on, so I'm going to ask you to stop sharing your screen, and we'll turn the podium over to Jennifer Arnold. Can see your screen.
Okay, can everyone hear me and see that in presentation view?
Yes, thanks.
Okay, excellent. So it's good to be here with all of you today, even if it's virtually. I want to start out by giving a shout out to my co-authors, Steve and Oswald, who those of you that know us well know that pretty much all of the work that comes out of our lab represents an equal contribution from both of us. And then a bunch of undergraduate student co-authors who contributed greatly to the collection of data on tern sleep and vigilance. So those of you that were here last year might remember that we presented a study introducing what we know about nocturnal sleep in terns and proposing ideas as to what might be driving sleep behavior. Today, I'm going to dive a little bit deeper into that, exploring the ecological factors that drive tern sleep and examining sleep patterns throughout the 24-hour cycle. So first, a little background. There's still a lot to learn about sleep in avian species, and what we do know has been largely influenced by our understanding of mammalian sleep. Research in birds, however, suggests that sleep functions in self-maintenance, cognitive function, and energy conservation. That said, the consequences of sleep deprivation may not be as severe as has been shown in some other species. For example, sleep deprivation in lab rats can lead to physiological impairment, and in just about 2 to 3 weeks of sleep deprivation, you can see mortality. And in fruit flies, sleep deprivation also similarly results in death. However, studies of sleep in birds, which are far and few between, can show a huge range of variation both within and across taxa. Within species, sleep tends to be strongly influenced by ecological factors, some of the ones that I've listed there. Although long-term impacts have yet to be studied, field studies are virtually non-existent, and what we know tends to come from very short-lived species. We do have some studies to look at. A study of sparrows suggests that songbirds can multiple studies of sparrows suggest that songbirds can at least tolerate some changes and reductions in sleep. So, for example, a study of white-crowned sparrows that was experimental showed that despite a 65% reduction in sleep during the migratory period, birds showed no impact to cognitive function. However, interestingly, those same birds, when it was not the migratory period, did show there was a strong reduction in cognitive function and result to sleep deprivation. So when I say we know little about sleep in avian species, we know even less about sleep in terns. There are some observational studies in Arctic terns where at the breeding areas with 24 hours daylight, about 3.5 hours of sleep were observed during the daily cycle. And in subarctic areas, during a 12-hour observation period, about 3 hours of sleep were observed during that time period. In this case, no one has looked at the full daily cycle, so we don't know if it was 24 hours, if it would be 6 hours, or if it would still be 3 hours. We recently published the first data on sleep for common terns. This publication by an undergraduate student, Deal et al., examined sleep behaviors and quantified sleep duration for common terns. And I'm going to start off giving you a little bit of background from that study. So that study, as well as the work that I'm going to talk to you about today, took place at 2 inland breeding sites in North America in the Great Lakes region, which are indicated there. And the first site was Presqu'île Provincial Park in Ontario, Canada. The second site was Little Island in Oneida, New York. And notably, these sites differed in that Presqu'île Provincial Park has high predator activity. It's a mixed species colony. There's rather large islands here where the birds are breeding, and the colony is about 100 pairs of terns. And Little Island had low predator activity, was a single species colony, much smaller colony, but much larger number of birds in the population. So at this site, we were able to see that terns tended to have similar sleep postures to other related species. So we saw a prevalence of back sleep, which is noted as generally being a deep sleep behavior, and front sleep pictured here. And at our site at Presqu'île Provincial Park, just one second. Okay. At our site at Presqu'île Provincial Park, when we looked more closely at the sleep, we saw that during the night, there was a lot of non-sleep behavior going on, and birds were only in this deep sleep, this back sleep behavior for a very short period of time. When we looked at Little Island, we saw a change in this. We saw much less non-sleep behavior and a much higher proportion of back sleep. Now, when we looked at this a little bit different way, by looking at whether eyes were opened or closed, and we did this, we looked at both these postures and whether eyes were open or closed, because in studies of birds that looked at brain activity, it was shown that the back sleep posture and the eyes closed behavior were indicative of them being in sleep. So what we see here is pretty similar. We see that only 64% of the night, the birds had their eyes open at the Presqu'île site, and that 17% of the night they had their eyes open at Presqu'île. So they were sleeping a lot. I'm sorry, I said that backwards. That 64% of the night at Presqu'île, the birds had their eyes open, and 17% of the night they had their eyes open at Little Island. So the take-home message from this was basically that birds were sleeping a lot more at the site that didn't have a lot of predation and was a larger colony. Oops. So this year, in this talk, I want to dive a little bit deeper into some of the causes for the differences between the colonies. So the aim of this particular study that I'm going to present today is understanding colony differences and putting nocturnal sleep in context of the 24-hour cycle. So I'm going to address the following questions. Are these differences that we're seeing between the colonies, are they consistent across years? Can predation risk explain the differences between these colonies? Is nocturnal sleep compensated for, perhaps by sleep during the day or sleep at roost? And are there any patterns that are different in sleep across the different sexes? So in terms of our methods, in 2019, we collected data at both Presqu'île and Oneida Lake. And photos were taken with infrared cameras, and we had 1-minute time-lapse intervals. And in this year, we trapped birds. Both members of the pairs were color marks. We had individually identified birds. In 2020, we only worked at Presqu'île, and photos were taken at roosting locations. We're just in the very early stages of analysis of this data. So the data I'm presenting here represents just 3 24-hour cycles in mid to late incubation, but the process was the same in that we were doing 1-minute time-lapse intervals. So I'm going to start off by addressing the first 2 questions. Are the differences consistent between colonies, and can predation risk explain these differences? So you'll remember this is just really a different way of showing what was going on in those 2 colonies. In 2018, we had a lot of vigilance at Presqu'île and very little sleep. And at Little Island, we had very little vigilance and much more sleep. And so several authors have suggested that the level of predation threat can be one of the things that is impacting sleep for birds. They've also suggested several other hypotheses, such as group size having an influence, whether it's a mixed species or a single species colony, and vegetation cover. But when you look at these hypotheses more closely, they're really all just things that modulate the level of predation threat. So in 2019, we were able to look at this predation hypothesis. We were very lucky to have a natural experiment occur. So you remember when I introduced the sites a little while ago, I said that Presqu'île was a site of heavy predation and Little Island was a site that very rarely had predation. But what happened in 2019 was that there was not really predation at Presqu'île, but at Little Island there was intense predation. And at this point, if you haven't figured it out from my pictures, I just want to note that our main predator, although there are others, is our black-crowned night herons that also nest at the Presqu'île site. Okay, so this was our 2018 data. So the first thing we wanted to look at these two questions of, were the sleep patterns consistent across years, and then test the predation hypothesis. And if we look at Presqu'île in 2019, we saw that the birds were actually even more vigilant in 2019, and they were actually sleeping even less. This is interesting because you'll note that I said that there wasn't really a predator there that year. However, we think that this is because there were some other things going on that year. First, some of these birds were on their fourth nesting attempt because we had such intense flooding that year that the birds kept getting washed out. But also, remember, this is a site that consistently over many, many years has had a lot of predation, and the predator is nesting at the site right alongside these birds. So even if the predator wasn't directly in the colony, there might have been the perception of predation by the terns. In relation to the predation hypothesis, though, what I think is maybe a little more interesting even is that when there was predation at Little Island, we definitely saw an increase in vigilance behavior and a decrease in sleep. And this was this increase was 3 times. But you will note that it still didn't go quite as high as it had at Presqu'île in other years. Okay, so the next question is lost nocturnal sleep compensated for by sleep during the day? So this is just a reminder of what the nocturnal sleep looked like. So remember, it differed between the two colonies. At Presqu'île, 79% of the night, the birds had their eyes open and they weren't sleeping very much, whereas at Little Island, they only had their eyes open 42% of the night. So very different between the two colonies at night. This is what the diurnal sleep looks like. I mean, the first thing you're going to notice is that these two pie charts look really, really similar. These birds are pretty much sleeping about the same amount between colonies during the day. The other thing you'll notice is that they're not spending a lot of time in that back sleep. And so, both of those things are kind of evidence that they aren't really compensating for this lost sleep during the diurnal period. We can look at this also with the eyes open, eyes closed approach, and you can see that they spend a very similar proportion of the day with their eyes closed. Okay, so then are they sleeping at roost? So at our roost, we found that the birds were pretty much only present at the roost at night. So the rest of the data I'm going to present refers to nighttime. And what you can see here is the percent of birds that were at the roost that had their eyes closed on 3 different dates. And here you have the percent of birds that were in back sleep on 3 different dates. And what I want to point out about these different dates is that on 6/4, there was no known predator active at the colony site. This work, as a reminder, was all done at the Presqu'île site. However, on 6/9 and 6/14, there was evidence of predation on both of those nights. And so what we do see here is that there does seem to be some sleep occurring at the roost, but that this sleep does seem to be reduced by predator activity. And as we're looking at this data more closely, I didn't put it in here, there's a suggestion that some birds could be getting, on nights when there's no predators, as much as maybe 3 hours of sleep at the roost, but on nights where there are predators, they're probably getting more like 20 minutes or less of sleep at the roost. Okay, so the final question we addressed was, are there any patterns among the sexes? And the simple answer to this is no. Here I'm showing the attendance of males and females at both sites during the 24-hour cycle. And on the other side, I have the vigilance during the 4-hour cycle. Now, we looked at this a lot of different ways. We looked at just during the day. We looked at just during the night. We looked at when both individuals were present at the nest, when only one was present at the nest. But in all the different things that we looked at, there was no difference between the sexes in their behavior at this point. So to summarize, and going back to my original questions, are these differences consistent between colonies? Yes, they are. So we did always see consistently more sleep happening at the Little Island site than at the Presqu'île site, but we did also see that environmental conditions were modulating that impact. Can predation risk explain these differences? It does seem that the risk of active predators does increase vigilance and reduce sleep. Is lost nocturnal sleep compensated by diurnal sleep? There's no strong evidence of compensation, but we do know that some sleep is occurring. What about at roost? Possibly they're catching up with some sleep at their roost. The data are still unclear because we're in the very early stages of analysis with that, but one thing we can say for sure is that predator activity does reduce the amount of sleep behavior. And are there patterns among sexes? No, we did not see that. So in conclusion, we need to first put this all in the context of the fact that little is known about sleep in birds, and that most of the studies that we have for birds come from short-lived species and captive birds, and that virtually nothing is known about turn sleep. Our study shows that there's a large variation in sleep within the same species, and that there's little compensation, that in the presence of predation, there could possibly be little compensation for this lost sleep. So if we kind of start to put all the numbers together in the time periods when there's predation, we could have the birds that are maybe sleeping about 40 minutes or so during the day. Maybe they're sleeping about 20 minutes at the roost, and they're not sleeping at all at night. So maybe they're getting an hour of sleep in their whole 24-hour cycle during these heavy predator periods. But the big question here is, how important is this, and does it really matter? So Metis, back in 1975, boldly suggested in his article in Animal Behavior that sleep must not be raised above the status of a spare time activity. In so much as sleep interferes with essential waking activities, it must be regarded as counterproductive. So in some senses, some people are thinking maybe sleep for birds is just some sort of extracurricular activity that they do. As humans, of course, it's hard to imagine that a K-selected organism could survive for long periods of time, which is what might be happening at some of these breeding colonies that are always subject to intense predation with so little sleep. But perhaps they can modify their sleep during this high stress time period in their life cycle. And you know, maybe during other times in their life cycle, they are getting more sleep. So certainly future studies need to address these points, and they need to help us improve our understanding of turn sleep, but also bird sleep in general, and help us to understand the consequences of prolonged sleep deprivation to birds during different phases of their life cycle, both in terms of physiology and cognitive function. I would like to end by thanking the large number of people that helped out with this project. As was mentioned before, whenever you have camera studies, there's a lot of people that are there helping out, but just thanks to everyone who's been involved. And with that, I'll take any questions if I have some time.
Brilliant. You do have time, and there are questions.
Okay.
Dave Moore, I think, jokingly says, do new parents ever get sleep?
New parents, I've been a parent for 11 years and I still don't get sleep, so.
Elizabeth Craig asks, are birds at the Presqu'île roost presumed to be actively breeding birds?
Yes, so that's a great question, and the way we identified the roost was with nano GPS loggers, so we know that those were birds that were actively breeding.
Jeff Spendolo says, how far was the roost site from the colony site, and was it also being used by non-breeders or failed breeders?
Okay, so there were actually a couple of different colony sites, and the roost site that we were looking at was not very far from it at all, like a few meters in a mud flat, not too far away. Whether there were non-breeders or failed breeders, we don't have that information, unfortunately, but there likely were some. Well, at that point, there probably weren't failed breeders, but there could have been non-breeders.
And your last question for right now is from Don Lyons. Do you suspect there's a relationship between how much sleep terns get and how much foraging they can do the next day?
Wow, that's a great question. And that's where, you know, as we're diving into the literature and we're learning that, you know, at certain phases in their life cycle, birds may be, at least short-lived birds, may be able to tolerate a lack of sleep. It might not influence their foraging the next day. Especially as we're seeing that there's not really like much diurnal compensation going on. They don't seem to be changing their behavior when they're getting less sleep the night before, so.
Brilliant. Thank you, Jennifer.
Okay, thank you. I'm gonna stop sharing now.
Yes, please. We do have another student coming up. So 2 more talks of this session, folks. Next up, we have Lauren Lescour. So Lauren, if you could share your screen. And Lauren is a student. I can see your screen, so take it away.
Perfect. So yeah, thanks for tuning in today. I'll be presenting on some of the data for my master's thesis at the University of Manitoba. Let's see if we can click through slides or not. Okay. So shifts in prey availability can be reflected in the diet of predators, although some species may be more or less affected than others due to a competitive advantage, such as morphological, physiological, and behavioral traits. And in marine systems, predators rely heavily on forage fish. So understanding how species interactions among predators vary in response to changes in their prey can indicate the ecosystem level impacts on these forage fish, which may be due to natural fluctuations, overfishing, and climate change. So my research was conducted on the northeast Newfoundland coast, and in this region, capelin migrate in high abundance during the summer to spawn. And this pulse resource transforms the coastal prey base into this superabundant capelin-dominated state. So with this huge abundance of quality prey, we expect to see a massive response from predators. For instance, during this time, migratory species such as whales and shearwaters arrive, and many predators are known to shift between prey, incorporating mainly capelin into their diet. And so perhaps expectedly, the chick-rearing season for seabirds in this area tends to coincide with this inshore movement of spawning capelin. Yet changes in ocean temperature and sea ice dynamics have resulted in this phenology becoming highly variable, where capelin spawning and arrival time are continuing to shift. So to represent this variability, I'll first orient you to this graph. So on the x-axis, we have the ordinal date, where 210, for instance, represents July 29th. So we're looking at the middle of the summer here. And then on the y-axis, we have our different years of data. And then the smaller black dots show the date range when hydroacoustic surveys were conducted to quantify capelin biomass, with the first and last survey date shown for each year. And then the red dot indicates the date of peak capelin biomass, with the larger the dot indicating higher biomass, like in 2014 here, and the smaller dots indicating lower biomass, like in 2016. And then the blue dots, which just for note don't change in size, indicate the first date of spawning, which is typically associated with peak biomass at least up until 2017. So if we think about this in the context of seabirds, in the green box representing peak chick-rearing for some of these species, this increase in food availability could happen outside of this really important and highly energetically demanding period for some birds, like in 2017. So this would force these birds to rely on other prey that are found farther away and in lower concentrations. So if capelin spawning and arrival continue to shift and change, there's the potential for a phenological mismatch with the chick rearing period, resulting in conservation concerns for these and other predators in coastal Newfoundland. So the years that we'll be focusing on include the years from 2017 to 2019. So as I've mentioned, capelin have become more variable, but we really see that occurring here during these years with these really large shifts in capelin spawning and peak biomass. So to shift gears for a minute, stable isotopes for diet studies of adult seabirds have become more popular as seabirds forage in distant locations that are not easy to observe, and they provide a method to examine diet using a non-lethal sampling process. And then delta values, which are the ratio of light to heavy isotopes, are typically represented on the biplot, which represents the isotopic niche, which has been used as a proxy of dietary niche. So we'll use this representation of diet to see how diet may be shifting among years. And then niche breadth is typically represented by the standard ellipse area, which is a proxy of the diversity of prey types consumed by predators. So for instance, species 1 has a much larger ellipse and would be foraging on a higher diversity of prey, while species 2 would be consuming fewer prey types. And then niche overlap, which looks at how overlapped the standard ellipses are, represents the amount of shared resources among species. So for my research, I'll be focusing on carbon and nitrogen isotopes, which are represented on the X and Y axis. And consumer tissues are enriched in nitrogen compared to their prey. So as you increase in nitrogen, you can get an estimate for the trophic position of the consumer, while carbon stays relatively the same from prey to predator. So you can get an idea of foraging location where carbon is higher in benthic dwelling organisms. So you can get an estimate as either inshore or offshore and as pelagic or benthic. So the main goal of my study was to examine how the isotopic niche measurements among 3 chicorine alcids, the Atlantic puffin, the razorbill, and the common myrrh, are responding to interannual changes in capelin biomass across 3 different years. And we hypothesized that the isotopic niche measures of all species would be influenced by interannual changes in capelin availability. So we predicted that when capelin availability is low, the isotopic niche breadth, so our ellipses, will be more spread out, and therefore birds will be foraging on more prey species, and there will be less overlap among species, so less shared resources. And then when capelin availability is high, we predict a higher reliance on capelin, indicated by this contraction of isotopic niche breadth. So our ellipses will be much closer together, taking up overall less space, and therefore foraging on fewer prey items. In addition, we predict there will be an increase in overlap where species are sharing the similar food resource of capelin. So our seabird research was conducted across 3 different islands, including James, South Cabot, and Funk Island. And to quantify capelin availability in the area, we conducted ship-based hydroacoustic surveys over capelin deepwater spawning sites. So we did this weekly during July and August in all 3 years, and that survey is highlighted by the green square. So it's important to note that although we only survey this small area, it does represent capelin in the overall study area, as it takes capelin less than a week to make their way west and then out north towards Funk. And then following the field season, capelin biomass was measured using EchoView software. So to determine the timing of capelin spawning, persistent deep water and beach spawning sites were monitored weekly or every second day for signs of capelin spawn, which was determined by the presence of eggs attached to sediment. And then we used this data to determine our years as either higher or lower capelin availability. We also collected blood samples from 20 individuals each year from common myrrhs, Atlantic puffins, and razorbills. And for this study, we focused on red blood cells as our tissue type of interest, as it represents the average diet over 3 weeks. So we identified potential prey types in the diet of these 3 species through observations and motion sensor camera traps of parental prey deliveries to chicks. And since isotopic prey values may change among years and season, we also collected prey each year in a variety of ways, including samples from fishermen, bottom crabs, and even misnetting of puffins. Our prey ended up including capelin, sand lance, and small fish, which are prey that were mostly less than 10 centimeters long and mostly incorporating larval prey samples. And then due to their similar isotopic values, we kind of grouped them together. And then stable isotopes were processed and analyzed for carbon and nitrogen using a mass spectrometer. And then there's a lot more that goes into stable isotopes, but I won't get into that too much today for you guys. And then for our capelin biomass results that were determined through our hydroacoustic surveys, I brought this graph back up, kind of focusing only on our study years. So as you can see, the timing of spawning varied quite a bit across years, and that's again shown by the blue dot, with the day of peak biomass not lining up as expected with the timing of spawning. So capelin were arriving into spawn, but not arriving en masse into the area until much later. And then blood samples from seabirds were collected 7 to 12 days around capelin spawning. So efforts were focused around this kind of peak chick rearing period. And then the key takeaway here is that we categorized 2018 as a high capelin biomass year, and 2019 and 2017 as years of low capelin biomass year, with 2017 acting more as this low capelin but late arrival, where capelin arrival and peak biomass are occurring close to or outside of this really important period. And 2019 is a low capelin but early arrival. So even though biomass was low, there's still capelin in the area during this time. So before I go into my results, I'll first reorient you to these isoplots, as you'll be seeing quite a few of them. So again, we have the nitrogen on the Y and carbon on the X. And for each prey, we have the mean delta value for the prey collected in the standard error bars. And then again, our 3 prey groups, which include capelin, sand lance, and small fish. And then each species will be shown with common myrrh shown in blue, razorbills in red, and Atlantic puffins in yellow. And then the dotted line shows the ellipse area, which represents the niche breadth. And then remember, we're looking at a proxy for dietary niche, so how these shifts in diets are showing up on these isoplots among different years. And then briefly to remind you what we expect to see. So during our year of low capelin availability, so in 2017 and 2019, we expect to see the ellipses more spread out and encompassing more space and having less overlap. And then during our year of high capelin biomass, so in 2018, we expect to see the ellipses closer together, values shifted towards capelin, and an increase in overlap as these species are foraging on a higher number of capelin. So we'll start off by looking at species level variation among years. I've also added all 3 years. So again, our 2 low years of 2017 and 2019 over here, and then our high year of 2018. And we'll start off with looking at just one species for ease of view and then add in our other species. So here I have Atlantic puffins and looking at their niche breadth. So again, the amount of space that they're occupying. And as predicted, niche breadth narrowed during years of higher capelin biomass. So there was a decrease in the diversity in prey in their diet as these birds foraged on a higher number of capelin. And the niche breadth was broader during years of lower capelin biomass, so there was an increase in the variety of prey in their diet. And then if we look at another species, so adding in razorbills, puffins and razorbills seem to be reacting to this change in capelin abundance in a similar way, with our high capelin biomass year, so 2018, having the smallest niche breadth, and then our low capelin biomass years for 2017 and 2019 having a larger niche breadth. And then lastly, if we add in MERS, MERS seem to be less variable in their niche. And we know that MERS in this area are capelin specialists. Well, they'll try to incorporate capelin into their diet kind of no matter what, which makes sense as they are single prey loaders, meaning they can only bring back one prey item at a time. So they really want to optimize on their foraging trip and bring back high quality prey for their chicks while also foraging for themselves. So potentially what this suggests is that puffins and razorbills are more flexible to these shifts in prey availability. And then if we look at niche overlap, so how the ellipses are overlapping and species are sharing resources, we'll start by looking at razorbills and MERS. And as predicted, there was an increase in overlap during high capelin availability. So the diet of these two birds were more similar, where they were foraging on mostly capelin. And there was a decrease in overlap during low capelin availability. So there was an increase in the variety of prey in these birds' diets. And then if we look at razorbills and puffins, there was an opposite trend. So unlike we predicted, there was a decrease in overlap during years of higher capelin availability. And during years of lower capelin availability, there was an increase in overlap. So potentially, razorbills are more sensitive to these changes in capelin biomass, or during lower capelin biomass, they're unable to compete with MERS for this less abundant prey item. And then for MERS and puffins, we don't see a similar trend or even an opposite trend like we saw previously. So niche overlap did not consistently increase during our year of higher capelin availability. So while all these species are foraging on similar prey items, they are occupying these different niches and incorporating capelin into their diet in a variety of ways. So if we want to look at how the assemblage, so all of these birds together are shifting among years, we can use 3 different metrics to look at trophic diversity, which reflects the amount of niche space occupied. So total area is, as it sounds, the total amount of space occupied by the assemblage, and the nitrogen and carbon range, looking at how these may increase or decrease among years. So if we look at total area among each year, in 2017, our delta values encompass the largest space, showing again that these birds were overall foraging on a higher variety of prey types compared to our other years. Then if we look at nitrogen and carbon range, again, we see the same trend with 2017 having the largest nitrogen and carbon range. So 2019 did not change like we predicted with such low biomass. So in other words, it did not follow the same trends as 2017. And this could be related to the peak biomass date occurring outside of the peak chick rearing period in 2017. So when we're focusing our efforts on blood sampling, the red blood cells would be representing an even lower biomass compared to 2019. So what we found was that our lower capelin biomass year was occupying more space overall, with our high biomass year tending to occupy less space overall. So in the end, we found that during the year when capelin availability was lower, birds were feeding on a wider variety of prey types, and there was a shift in interactions among predators with these shifts in capelin biomass, with MERS being the exception, where they will incorporate capelin in the diet during high and low capelin availability. So despite these fluctuations in prey, it seems that these birds may be able to tolerate these climate-derived changes in prey availability. But at what point is the point of no return for some of these birds, especially if MERS seem to be specializing in capelin? What consequences could capelin arrival and biomass have on these birds' reproductive success, where, for instance, they may have to spend more time away from the colony and increasing predation risk to their chicks? So we were fortunate enough to be able to go out this last summer where we collected capelin biomass data and blood samples from these 3 species of birds, which we plan to incorporate into this data set. And we had a very interesting summer with what seemed to be very low capelin numbers. So I'm really interested to see how these metrics will shift with this kind of more extreme summer that we had. We also plan to pair some of this diet data with GPS tracking information from razorbills from 2019 and 2020, which is really starting to take shape as we explore this data, and I think will tell a very interesting story. And then lastly, I just want to acknowledge the Davern Lab and the crew of the Lady Easton 3 for their help in collecting all of this data. It really takes a team. And with that, if we have time, I'll take some questions.
Brilliant. Thanks, Lauren. We do have 5 or 6 minutes for questions if there are any. Our first being from Rasa says, what were your sample size of capelin and other fish? Just standard deviations look quite small in the bioplots.
Yeah, so we had 10 capelin, 10 sandlands, and then not entirely certain on the small fish, but I want to say similar numbers to that. So we tried to not overwhelm our kind of work that we had to do with all of the stable isotopes, but still get a good representation of the fish that were out there.
The follow-up question to that is, are you going to use mixing models to calculate diet proportions?
Yeah, so that's our plan with the 2019 and 2020 red blood cells from our razorbills to kind of get an idea of how the diet is shifting with these changes in tracking information. So the distances and where they're going to forage. So that's in the next steps, yeah.
Beautiful. I was wondering if you had information on how successful nesting was in these 3 different years. So you've looked at diet and how that responded to prey availability, but what was the nesting response like?
We don't have too much information on that, and I know Bill Montevecchi, who works on Funk Island, will know a lot more about what's going on with the MERS on Funk. I can only really speak to the razorbills and a little bit to the puffins, as the puffins kind of nest later in the season, so we don't really get to see that fledge date. But with razorbills in at least 2019, they seem to be doing fairly well despite these low capelin numbers. But I can tell you with 2020, where we had extremely low numbers, where we didn't really see the shift to capelin, that their numbers, it looked like their fledging numbers and reproductive success were extremely low. And we weren't seeing a lot of fledgeable chicks, which was quite heartbreaking, but kind of, you know, a whole part of ecology in the study and what we're looking at. So, yeah.
Liz Craig says, great talk. It seemed like there was a fair amount of variability in the timing of capelin availability. Forgive me if you've already addressed this, but do you have a sense for factors influencing the timing?
I think there's a whole slew of factors that I am not 100% knowledgeable on, but we know that changes in kind of sea ice dynamics, which are influencing the water temperature, will change kind of development for reproduction for these fish. And so that can have an influence on when they arrive. Temperatures in the water will also be a big influence. So when it's too hot, the fish won't want to spawn because their eggs will kind of just bake in the water. And so I think these fish are probably reacting to all of these changes that'll influence when they arrive and when they spawn, if that makes sense, yeah.
Yeah, it was interesting to see in your later years how the spawning and the peak spread out so much.
Yeah, we don't entirely know why that's happening right now. We have, I guess, some thoughts on it, where in the past, capelin would come in early in July and have this short, small spawning event and then like retreat, and then there would be another flow of capelin that would come in. And so maybe they're reverting back to this kind of previous behavior that they had, or we don't 100% know. It's quite worrisome, at least for the birds and other predators in the area with what's happening with capelin, yeah.
Don Lyon says, does their larger niche indicate that puffins are more flexible generalists or less capable foragers and competitors? In other words, are puffins less capable of getting capelin than MERS and razorbills?
So there's a potential for that, yes, because they are smaller and so they can't travel as far and they can't dive as deep as MERS and razorbills can, especially MERS. And so there's a chance that they aren't able to compete with those other species. We also know that they do bring in a lot of larval fish. So when capelin collapsed in the 90s, I think there was this huge shift with puffins. And so I think they kind of shift towards these larval fish because you see a lot of that in their diet when we're observing them. And so forgot where I was going with that. So you see the shift with towards that, and maybe that's just been a continuous trend since capelin have become so variable and so low.
Do you ever see evidence that murres switch away from capelin, or do they just have a poor nesting year when capelin aren't good?
I wish I knew more, because we don't spend a lot of time on the murre colonies. I don't have as much kind of insight knowledge on what's going on out there. We did see a lot of different prey that were coming in this last year in 2020, so they might be having to shift and be more flexible as capelin become more and more variable. So there's a chance that they will have to kind of learn and change their behavior as well.
Very good.
Thank you.
It is about time to move on. So thank you, Lauren. And I'll introduce Nathan Dubrow, who will give our last talk and last student talk of the session.
Hi, let me share, hold on.
I can see your slides and I can hear you. So thank you. Go ahead.
Awesome. So my name is Nathan Dubrow. I'm an undergraduate student at College of the Atlantic in Bar Harbor, seeking my BA in human ecology. So this summer I decided to conduct a preliminary study on leeched storm petrels in their weight restricted lives. So this is just kind of a basic thing and could continue on in future years as other students have similar interests. So let's do a little bit of background, right? I'm sure a lot of us know, but what is a leeched storm petrel? So it's a small seabird in the order Procellariiformes or tube noses, which includes others such as shearwaters, albatross, and true petrels. And these, like many other Procellariiformes, nest in underground burrows. So for these, as well as many other species, life revolves around food. They must consistently forage for food in order to maintain themselves, such as when they're not breeding, they move to lower latitudes to find places where food is more abundant. So food is stored as oils obtained from the prey, which is then converted into energy for use when they need it. So these birds feed offshore and primarily take zooplankton from the ocean surface, as well as any other small fish which may be available that is high in fatty oils. In this species, they breed like many other Procellariiformes on predatory mammal-free islands. And although while there are other predators such as gulls on the islands, the petrels have adapted to get around this by leaving and entering the colony at night. So they are more or less unseen by the gulls, which are typically resting during the nighttime. And so when they're nesting, and during periods of incubation, one of the pair of storm petrels fasts while the other partner feeds, and the other partner collects important fat reserves through oil collection for when it comes back when it's its turn to incubate the egg. So more background on the incubation period. So they only lay one egg per season, and the incubation lasts between 37 to 50 days. And so this long period of incubation, as seen in many Procellariiformes, represents the slow development of the embryo within the egg. And so due to this long period of incubation, adults must stay within their burrows for consecutive days without feeding, while their partner is out over the ocean collecting food and gaining weight. So they must use their reserves while they're in their burrow, and in doing so, they typically lose the weight that they have previously accumulated as a result. So let's look at the study location. So first, on the left is the Gulf of Maine. You can see Massachusetts, Maine, Nova Scotia on the right, kind of give you like a geo-reference. And then in the center is zooming in to where that red dot is, which is coastal Hancock County. And then you can see College of the Atlantic is the black dot in the center image. And the red dot is Great Duck Island. And then on the right is zoomed in all the way to Great Duck Island on the bottom with Little Duck Island on the top. So first, the scope of the study, I wanted to look at the decrease in weight of the adults as they're incubating their egg, as well as the increase in, sorry, that's supposed to say weight of the chicks. So first we had to find burrow locations. So on the left is Great Duck Island zoomed in with 10 burrow locations, which were originally located and were found to have adequate access for an arm to reach into the burrow and collect either the adult or the chick. So we only chose 10 burrows because it represents 0.0004 to 0.0006 of the total population of the island. So this is a fairly low number because we understood the high percentage of abandonment when storm petrels are harassed, you could say. So 3 of those 10 ended up abandoning rather quickly, while 1 of them had intermittent attendance, which is known in storm petrels as egg neglect, where they is regularly occurs that they value the survival of themselves over their eggs. So if their partner takes too long to come back and incubate, then they will leave and feed themselves rather than continue to lose weight. So methods for looking at the adult weight loss. First, as I said, we found the accessible burrows that were easily grubbed and could yield successful results for each attempt. And then the burrows were GPS mapped and marked, and they were marked with a pink flag along with the number, so we could easily find it each time we went out to weigh the birds. And then we banded each individual as well. So the first individual that was in the burrow originally was banded, and then as the second individual came to the burrow to incubate the egg as a parental switch, then we banded that bird as well. So then the weights for each adult were then measured each day, which is, yes, a little bit of a lot of harassment, but it ends up, for such a small percentage of the population, we found it to be all right. So we weighed each of these adults for a full cycle. So basically a singular bout of incubation for each individual of the pair. So first, one half of the pair might incubate for 4 days. And then, so we got that data right there. And then the next individual might incubate for 4 days as well. So we got just this full cycle of incubation weight loss for each one. And this was done by placing the petrel in a cotton bag and measuring the bird with a Pasola 100-gram scale. And then, so here's just a graph showing the individual average weight of each storm petrel studied. And while it's noted that females typically weigh a small percentage more than males and are typically just a bit larger, it's difficult to determine the sex of these birds, so that wasn't included in the study. Nonetheless, of each pair, one individual appeared to average heavier than the other, although, as you can see in 14, they were just about the same weight. So overall grams, when we're looking at overall results, when we're looking at the gram in terms of grams, each individual each day lost grams, although it varied by the day. Sometimes there was just a small percentage, while others was a small amount of grams. Other times it was a lot more. So as you can see here, when there's an apparent increase in weight, such as where the red arrow is pointing, the first up on the top, that's basically showing the time period when the adult is feeding out at sea and we're unable to get the daily weight increase. But when the points appear to go downwards, that's the time period when the adults are incubating and thus losing weight. So now if we look at the weight loss as a percentage, there was a lot of variation. So it ranged from the lowest point where the red arrow is pointing to approximately 1.98% of weight loss in one day, to where the black arrow is pointing to 6.77% weight loss in one day. And the average for this was approximately 4.8% loss per day, which this is, a lot for any animal, especially for a tiny little bird like this. So now if we look at weight loss percentage, weight loss of grams to percentage as a comparison, we can see that so this is just a subset of data looking at a particular cycle of two individuals in a singular burrow for burrow number 1. On the left is the gram daily loss, and on the right is the percent daily loss. And so for burrow 1 on the left, for individual 196119753, there was in the span of 2 days, the individual lost 4 grams, while for individual 196119747, the individual lost 2.5 grams. And then it looks a little bit skewed. So if you look at the percentage loss, I'll just say for 19753, this individual over the span of those 2 days lost approximately 7.7% of its body weight. And while the individual 19747 lost 6% of its body weight in those 2 days. This shows, according to this, that body weight lost is not exactly proportional to the original body weight. As you can see, the individual 19753 originally weighed less at 52 grams at the beginning of its incubation cycle, yet lost 7.7% of its body weight overall in those 2 days. Whereas 19747 originally weighed 58 grams, so 6 grams more than its partner, although it only lost 6% of its body weight over those 2 days. So why don't we look at a second one so we can figure out that burrow number 1 wasn't just an outlier. So this is for burrow number 5. And you can see as for band 1961, 1967, 19767, there is a 2 gram loss in 1 day. And for its partner, there was a 2.25 gram loss in 1 day as well. So if you look at the percentages in that 1 day of loss for first individual 19767, there was a percent loss of 3.57% of the body weight, whereas for 19751, there was a 4.5% loss of body weight in that 1 day. And once again, this was the individual that weighed less originally, still once again lost more body weight as a percent than its partner, which originally weighed more. Now let's look at the chick portion of the study. So for this, I also took a subset of the previously marked burrows, which had chicks, so only the burrows which had chicks that actually hatched, because some of them still hadn't hatched just due to the time period at which we were out there. So for this, we put up game cameras to assess the parental visitation at night. And unfortunately, some of those game cameras didn't work as well as we'd hoped. So I only took a subset of 2 different chicks. So for this, we measured the chick weight every other day, so that it gives the time to not have as much stress occur to them. And we also, once again, used the Pasola 100 gram scale. So first, let's look at the gain in grams over time. So it's shown at the burrow. So in burrow number 1, up on the top graph, there it shows that on scatter plot, there are more visitations for burrow number 1 compared to burrow number 14. And this also showed, if you look at the graph below, at the grams increased per day, it correlates into where the more visitations brought higher weight gain per day. And so as you can see in number 14, the weight actually decreased towards the end of the field season, and this just seemed to be a fewer visitations towards the end of the study. But we couldn't exactly see whether the increase, if the percentage if the weight continued to increase, or if it continued to decrease, because we just weren't out there for the time period to see if there was any change in that progression or decrease in weight. So if we look at the weight gain as a percentage, it showed that weight gain is fairly exponential in chicks, owing to nearly 400% increase throughout the study, at the end of the study. While daily increases ranged from about 11.76% to 35.3% gain, the average was about 18.3%. So this shows that chicks just happen to grow a lot quickly. They happen to grow quickly because when their parents are bringing them food, it's basically depositing the oils that the parents collect to the chicks, and then the chicks kind of store that until they use it for metabolic processes, or eventually use it to grow other feathers in size. So as a discussion as results, so basically just going back over the results, it's shown that evidently adults lose a significant percent of their body weight each day, which remains. So we see this, the average loss of 4.8% per day, and we see this without apparent adverse effects as they do it so regularly. It's kind of a wonder of how exactly they do it. As we saw, once again, the minimum weight loss was 1.98%, and the max was 6.77%. On the chicks, on the other hand, gain a significant percent of their body weight each day. So the average weight was 18.3% per day, while the minimum was 11.76 and 35.3 respectively. This begs the question, however, at what rate do the oils get provisioned into their growth compared to those used to maintain homeostasis? Because during the period of data collection, we didn't see any significant growth in feathers or anything. It was mostly just a chick getting showing a larger amount of oil in its stomach just by seeing the approximately the stomach mass, or looking at the just basically by direct observation. So further studies that could potentially would help to unlock more of this would look at the percentage body weight lost in relation to initial weight. So as we saw, the initial weight with individuals that had initial weights that were lower, they actually lost more a percentage of their body weight each day. So it's kind of interesting to just note that and see why is this going on and maybe continue studies further. And then look at the weight loss percentage differs, how the percentage of weight loss differs among sexes. Because males are just barely smaller on average, we could, think maybe those ones that weighed less originally are the males, but we don't know that for sure. And it would be good to go back and actually explore that further to see and kind of look at to see why this is occurring. And it would also be great to explore at trade-offs of adult weight gain during feeding at sea and subsequent weight loss in relation to chick growth in the burrows. So, as the adults are feeding in the ocean and how much they weigh when they come back, and seeing if what they feed their chicks, how much of their weight loss, how much weight they lose, and see if there's any correlation of the weight lost to the weight gained by the chick. There's actually, I believe, a study going to occur next year, hopefully up at Kent Island, looking at that using sensors, which this summer Robert Mauck brought some stuff out there to test in order to look for grant money to see how it was working in order to request further grant money to further that study. So hopefully it was kind of in proper timing of different studies to look at, data being collected now and seeing what can actually go further with a study like this. So that's all for now. I'd like to acknowledge College of the Atlantic for allowing a field season this summer, which originally wasn't going to happen at all. But Darren Collins kind of pulled some strings along with John Anderson, who's made this possible with talking, Darren and getting everything on board, as well as going through the Drury Fund just to get us money to live out there. I'd also like to thank Adeline Huckins for helping take data and moral support while on the island, as well as Dan Gaddy for help with data analysis over the last few weeks, and Aya Kumagai for guidance on working with storm petrels. And she was on their last year working with them as well, and we will hear from her later tonight, which should be cool. And I think that's it. There's some references. Yeah.
Thank you, Nathan.
Mm-hmm.
So we do have a couple minutes for questions for Nathan, and then I will say thank you to the audience and open it up for all of the speakers. So do we have any questions right now for Nathan? While people are typing in the chat, I wanted to ask, Nathan, you mentioned that abandonment is really high. Are you talking about abandonment at your site in particular, or is that typical of petrels? And if it's for your site in particular, do you think the weight patterns you observed apply to individuals on other islands?
Well, so the abandonment that I was referring to typically has to do with high disturbance levels. So that's why looking at birds daily will cause that high disturbance, and then the adults, if it's not too far along in the incubation process, then they're just going to kind of give up and leave. So that was kind of the thought there, which has been seen on other islands as well.
Okay. So you think the patterns you've observed in weight would apply to, for example, Kent Island as well?
Yeah, I would presume so.
Susanna says, is it possible a portion of weight loss measured in adults can be attributed to possible stress of being handled daily?
Yeah, I mean, that's definitely something that has to be taken into account. I suppose if you do a consistent amount of handling each day, then the percentage should the amount of stress should be similar, although it's kind of up for debate, I suppose. But then the weight loss would kind of just correlate with the amount of stress being seen in the individual.
Okay. Any other questions for Nathan? We have reached the formal end of our session, so I'll remind everyone that The next session starts in 20 minutes. It's about climate and conservation. So we've got another great set of speakers, and there's an international session happening as well this evening. So do tune in. I'm happy to stick around for as long as we want now if any audience members have lingering questions for our panelists that they didn't get time to ask earlier. But otherwise, I'll say thank you for coming, and thank you once again to our speakers. Okay. And Danielle's reminding us, don't forget to order your conference merchandise. There's a website. It's t-shirts, but it's a bunch of other things too with our logo. You can pick and choose what you want. Nathan, if you're still online, Jenna has a question for you.
Yeah, hold on a second, sorry.
Jenna, are you there?
I'm here, but I have really poor internet connection, so I used the chat.
I was just sorry. I was just gonna say hi.
See, I don't know personally right now if petrels do have a linear growth rate. Maybe definitely something to check out more.
Thanks, Lauren, for presenting. Nathan, how far along in your degree are you now? Is this your third year, fourth year?
My fourth year.
Woo! Is your senior project related to Great Duck, or is it something else?
It hasn't been completely, I don't have a full total idea exactly of what it's going to be, but it very well likely might encompass this, but it's also from Gannett data that I took last summer 2019 on Mount Desert Rock and presented last year as a poster.
Cool, right. I remember your poster.
Mm-hmm.
Was this your first conference talk?
Yeah.
Congratulations, that's awesome.
Thank you.
You did a good job.
Thanks.
Nathan, just out of curiosity, have you heard anything about if anyone's tried to use like artificial burrows for petrels? Because I know we do that with the rhinoceros auklets, which makes it a lot easier to monitor them. And I know there are so many burrows on Great Duck that are just impossible to reach. And I was curious if petrels might take to artificial burrows or not.
I feel like they wouldn't, because they typically dig their own burrows and then they use their scent to refine them. And I feel like that would be a rather difficult thing to initiate. I mean, it's definitely worth a try. I know on Kent Island, they just kind of change the shape of some of the burrows to make it easier to retrieve them. So that's definitely a thought, because, I mean, once they have a burrow, they'll probably likely to return to it, even if it's a slightly different shape. But I think if you made a burrow, it'd probably be less likely to actually take to it.
Nathan, have you been up to Kent?
I've not.
But it sounds like you're in touch with some of the people there, at least.
Mm-hmm. Yes. Robert Malk, who from Kenyon Island, who works a lot from Kenyon College, who works a lot with Bowdoin and Kent Island, came out to test some things with our storm petrels because they didn't have a field season this summer.
Oh, okay.
So in order to test the sensor and seek further grant money, they came out to our island with proper precautions, and we just grabbed a couple of petrels and put it through the sensor.
Lucky you to get to meet him.
Yeah, it was fun.
Yeah, I've heard his name a lot, but I never got to see him.
Mhm. Well, I think I have to go. I'll
Yes.
probably join on later, but I got a class.
Good luck.
So
Thanks
yeah,
again.
thank you everyone.
Jenna, how's your research coming?
It's coming along. I wanted to present for this, but I didn't have any finished results, so I thought it'd be better to wait for next year so I have something a little bit more complete. But
Did you get a field season or was yours canceled?
I got a field season, but it was pretty crazy. You know, we usually have a team of between 8 and 14 people, and I got to go out with just me and 4 interns, some of them very green. And so trying to manage population monitoring for like 4 different species with just our crew was a lot.
I bet.
But we did it. Yeah. So unfortunately, I didn't get to focus a lot on my project in order to make sure that we got the data that we needed for Middleton. But
Yeah.
I mean, I think the information that we got was great. I mean, the kittiwakes actually did a lot better this year than they have in past years. So that was really exciting to see.
Why do you think that is? Is it food or something else, or you have no idea?
If I had to guess, like totally guess, this is not based on anything that I've like found, but they had a really harsh winter, I guess. And so I'm wondering if maybe the really cold winter kind of made for better foraging conditions in the summer, you know, helping keep water a little bit cooler and helping make fish more available for the kittiwakes. Because we see in years where there's really high sea surface temperature, those are the years that they tend to do worse. So that would be my out of nowhere guess.
Do you mean the sea surface temperature like affects how you said more available? The fish should be more available. Does the sea surface temperature affect how far down in the water column the fish hang out? Is that what you're saying?
Yeah.
So if it's a cold winter, then it's the surface temperature is cooler and the capelin can come up higher.
Mm-hmm. Okay. That would be my guess.
Yeah. No, it's cool. I don't know much about your system, so it's interesting.
Yeah.
So which are better, kittiwakes or herring gulls?
I still really love herring gulls.
Yes.
I think that they're slightly more charismatic, and I think that the kittiwakes are actually more vicious.
Really? I don't know that I've never seen a kittiwake that wasn't like way off on the horizon, I think. So I'm jealous.